Next, let us discuss the migratory cells of connective tissues. First will be the mastocyte. The mastocytes, also known as the heparinocytes. These cells contain granules, but first let's start with the cell nucleus and the adjacent organelles such as the endoplasmic reticulum and Golgi complex. However, we should mention the granules in the cytoplasm that contain some active substances such as heparin and histamine. And upon stimuli such as binding of some antigens to the type or class E immunoglobulins on the surface, a process called degranulation is triggered. Where the content of the granules is released into the extracellular space. So we have the nucleus, the endoplasmic reticulum plus Golgi here, and the granules. The granules contain substances such as histamine, and histamine granules cause the increase of permeability of blood vessels. Together with the increasing the diameter, which is called vasodilation. So the surrounding blood vessels respond on the presence of histamine by increasing the spaces among the endothelial cells and by increasing the diameter and by relaxing the smooth muscle that surrounds the blood vessels. Both mechanisms allow more fluid to escape from the blood plasma into the extracellular fluid, causing edema and contribution to various phases of inflammation. There are also heparin granules. So these are blood vessels, okay, undergoing the increase of permeability and vasodilation. Let's go back to the heparin granules. Heparin is a substance that is a natural glycosaminoglycan and it's an anticoagulant. So this anticoagulant agent impairs the blood clotting. Other famous migratory cells of connective tissues are the macrophages. So, macrophage originate from circulating blood cells called monocytes. These are the largest white blood cells that occur in the peripheral blood. And these blood monocytes are entering the tissues where they become macrophages. They mature into macrophages. Macrophages are huge cells with many cell processes, irregular processes, feed, and they are wandering through tissues and searching for antigens they might eat. The cell nucleus has also a highly irregular shape. There is also endoplasmic reticulum, Golgi complex, and one of the organelles typical for macrophages are lysosomes. They have very well developed lysosomes, membrane-bound organelles filled with enzymes. So lysosome with enzymes. When the macrophages encounter some particles, which might be dead cells, cells infected by viruses, necrotic cells, exogenous pigments, etc., they have a tendency to surround the cells with their processes and swallow the particles. So it's the endocytosis happening here. And endocytosis of larger solid bodies is called phagocytosis. Result of this endocytosis or phagocytosis is an endosome called phagosome. They merge with the lysosomes into phagolysosomes, where most of the content of the phagosome is chemically destroyed by various mechanisms. Hydrolytic enzymes, reactive oxygen species, such as superoxide and peroxide ions, etc. So this is a phagolysosome. However, fragments resulting from this partial autolysis are exposed on the surface of the macrophage. If this occurs in the context of so-called MHC class II molecules, where MHC stays for major histocompatibility complex, MHC class II molecules. You might know the MHC in human also under the abbreviation HLA, which stands for human leukocyte antigen. So together, the antigens exposed in this context work as an activating signal that helps to activate other immune cells. For example, T lymphocytes. So if the T lymphocytes are equipped with a receptor able to bind to these antigen fragments, there is a There is activation of these cells in the sense of proliferation and production of various substances such as cytokines, interleukins. They may also stimulate other immune cells such as B lymphocytes. Again, with a specific ability to bind with these molecules. And these activated B lymphocytes differentiate into other cells of connective tissue, namely the plasma cells. A plasma cell is the producer of immunoglobulins. That's the major effector mechanism of so-called humoral immunity mediated by antibodies. The nucleus is very found in an eccentric position, and the distribution of the chromatin is characteristic. It's called a clock phase or cartwheel chromatin. There is a huge rough endoplasmic reticulum responsible for the basophilic properties of the plasma cell, and these newly produced immunoglobulins are prepared for exocytosis and the Golgi complex, which appears often as a lighter region in the cell. So we have this clock phase or cartwheel distribution of chromatin in the cell nucleus. We have extremely well developed granular endoplasmic reticulum with the ribosomes on it, where the proteins are produced. Therefore, the cells are strongly basophilic. There is the Golgi complex there, and these plasma cells are producing immunoglobulins. Abbreviated very often as Ig, and you know these as antibodies. The major point of that branch of specific immune response is that the antibodies are able to bind the antigen fragments, the fragments of the very antigen that initiated this whole cascade of processes, which has been simplified for our purposes.